Let's start.
Hello, everybody, and thank you for joining today for the Molecular Connectivity Online Series, or MCOS. I'm Matej Peronik from University Medical Center Ljubljana in Slovenia, and today I'll do the introduction and help with moderating the discussion. For those of you joining us for the first time, the Molecular Connectivity Working Group is a community of researchers dedicated to advancing molecular imaging as a tool for studying brain connectivity. We plan this series as a platform to exchange ideas and promote the latest research in the field. For further information, you can visit our website, subscribe to our newsletter, or follow us on Twitter and LinkedIn for updates. And before we begin, just a quick groundkeeping note. We are recording this session and we'll upload it to our YouTube channel. And to protect our speakers' research, we kindly ask that no private recordings be made and that this content is not uploaded to any third-party websites. With this out of the way, now I'm honored to introduce today's speakers. Today's speaker, Dr. Prashin Unadkat. He is a great friend and a chief resident of neurosurgery, just about to finish at the Zucker School of Medicine at Hofstra, Northwell. He is an incoming fellow at Baylor College of Medicine in Houston. In his clinical practice, he specialized in advanced image-guided surgical treatments for movement disorders, epilepsy, and psychiatric conditions. Alongside this, his research focuses on using brain imaging to improve surgical planning and predict how well treatments will work, especially when it comes to deep brain stimulation. And today, he will present on preoperative network activity and how this predicts the response to subthalamic deep brain stimulation for Parkinson's disease. So please join me in welcoming Dr. Unadkat, and I give you the floor.
Thanks so much, Matej, for that kind introduction. Me and Matej know each other from his time in the US. So I'm really excited to be sharing my work that came about during my time during my PhD thesis work. And just to get things kick-started, I'm going to kind of break the talk up into like a couple of sections. So the first section will be just to describe the problem, why I chose to study this and what I found interesting about it. The next will just be the approach that we took and to biologically validate this network that we identified. And then the third section would really be focusing on, now that we have this network, what could its potential utility be? And then we'll kind of open the discussion up to any questions that people may have. So just to get started, we have this project published in the Journal of Neurotherapeutics a year and a half ago or so. And it was basically to understand, the first premise was to understand what are the brain regions that get modulated in response to very focal stimulation of the subthalamic nucleus. We know that DBS to the STN is extremely potent, but why does it work so well? What regions are being modulated and how can we make it better? Because there is some variability when it comes to treatment responses. And how do you even identify who's a good candidate? And I'll go into that a little bit. But first, let's just take one step back for a brief second, and I'm sure people don't need a detailed introduction, but why do we do STN DBS for PD? So we know PD has the classical three symptoms, and these three symptoms are the ones that typically respond very well to DBS. So this bradykinesia, rigidity, and tremor. Tremor particularly works very well, so does rigidity. Bradykinesia sometimes takes a little bit of a fine-tuning to be able to get a good enough response. Patients typically experience improvement in gait disturbances. Dyskinesias improve significantly, and so do dystonias. And there are other targets that we can target for PD when you have predominant dyskinesia and dystonias, but that's beyond the topic that we need to discuss today. The other main symptoms of PD that do not involve this classical motor triad, they don't usually respond, and they're not relevant to the talk that we're discussing today. So once someone has a diagnosis, and there's been a lot of work on how do you diagnose someone with PD, the focus becomes symptom management. Till date, we don't really have treatments that reverse PD, but they help manage the symptoms. And so we talk about the classical dopamine replacement. Polypharmacy becomes a strategy for patients experiencing other side effects as well as other symptoms. And then finally, the thought process is deep brain stimulation comes towards the end when motor symptoms are not controlled well enough with dopamine. So it's important to identify who is going to be a good candidate. And we still don't have very objective markers to determine that. Then there comes the problem about Parkinson's disease being an emerging pandemic. The number of patients being diagnosed every year are increasing exponentially. And that becomes a factor. DBS is an expensive procedure. And not only is the surgical related procedure expensive, but also there are other healthcare costs related to it. So if you're doing a treatment that's only symptomatic and you don't land up getting the benefits you do, on top of that, it is an expensive procedure to perform with some surgical risks associated with it. You really want to try to be sure that the patients will experience a benefit that meets their expectation. And that's important. I want to really highlight that DBS is really meant to fulfill the expectations that patients have. And it's not really reversing their underlying condition. So for that, we need the treatment to be effective and meaningful, economically justified, and adjustable because disease progresses, and you need options to be able to make therapy better as the disease gets worse. So DBS for Parkinson's involves implanting an electrode either unilaterally or bilaterally, and typically the three targets associated with PD: the STN, the GPI, and the VIM. The VIM is specifically reserved only for patients with tremor-dominant Parkinson's with no significant symptoms of bradykinesia and rigidity, and it's more used for patients with ET. But in PD, some patients will get VIM DBS as well, and the current is delivered on a chronic adaptive basis. And so the challenge lies in these three factors. And, I don't want to spend too much time discussing this, but when patients start to experience fluctuations, they start to have dyskinesias, and they start having a complete loss of efficacy, that's when the patients get identified for DBS. And the way we identify patients for DBS right now typically falls down, especially in the United States, in terms of insurance approvals, falls down to the levodopa challenge test. Now, this was designed specifically to help differentiate the atypical Parkinsonian syndromes from PD. And yeah, when you have a response of about 30%, that's when patients are deemed candidates. But can we do better? The way I think about DBS, and I think this is important because that's what the entire talk will kind of focus on, is I think about DBS in terms of an optimal outcomes from DBS involving three main things: patient selection, and that's what's going to be the focus of our talk today. For that, you need accurate diagnosis, levodopa responsiveness, cognitive status, and disease severity. All these factors play a role in identifying who's a good candidate. Electrode location. There's a lot of research going into this right now, and we kind of touch on it in the talk a little bit later. But electrode location matters. We've gotten pretty good at putting the electrodes in the right spot, and you need to have an accuracy of less than 1 to 2 millimeters from the set target to get an appropriate response. But I think that's a problem that we're solving very well. And then finally comes the stimulation parameter titration. Electrodes are getting more and more complex. There's more and more contacts. There's more and more stimulation options. And so sometimes it can take almost a year for patients to get fine-tuned on their final DBS parameters that give them a good relief. And then the disease progresses. So you come back to titrating things again and again. Again, I think with a more objective marker that can measure the effects of DBS, you can probably hit the patient selection option better and the stimulation parameter option better. And so the goal of what I'm going to discuss today is trying to find that optimal target between these two factors that contribute to DBS outcomes. So let's talk about STN StimNet. The experiment that we designed for this was involving predominantly FDG PET data. We took PD patients that had been diagnosed with Parkinson's disease and were implanted with bilateral or unilateral STN DBS. They came in on 2 consecutive days. The order was randomized, but on day 1, they had an FDG PET scan done in the off stimulation phase, so the DBS effect was turned off. They typically had about a 24-hour washout period for DBS off, so by then most of the biological effects are gone. Their levodopa doses were withheld to not confound the analysis. And on day 2, they underwent sequential imaging, first with O15 PET to look at blood flow, and we'll talk about why this was important and somewhat novel, followed by getting an FDG PET scan on the same day in the DBS on state. To identify this network, and I come more from the clinical realm, so I'm not going to go into the details of this, but in brief, once the images were pre-processed, we used a software package that I think a lot of people might be familiar with, the ordinal trend analysis. I think Dr. Habeck has been on this series before and spoken about it, and he had written the first paper about this. But it basically is a form of supervised PCA that looks at the only thing that's given to the algorithm is the condition, that is the off versus the on. No clinical factor such as the amount of motor improvement is given to the algorithm for network detection. So in a way, it's pretty unbiased. Once we identify a network pattern that fulfills criteria, you can see the topography of the pattern, which regions are heavily modulating and separating the two conditions. And finally, you can calculate the network expression of the pattern in each of the conditions on a subject by subject basis. So that network expression score becomes your biomarker. So first, for this, we took 10 patients we took 10 hemispheres, from 5 patients that had STN DBS. We selected the patients that had the best therapeutic response. That means their motor symptoms improved well. So in a way, you're kind of feeding the best subjects to the pattern, but it was also to try to isolate effects of stimulation that were optimal and something that we were interested in. And what we found is that the network expression between the off and on condition increased in 9 of the 10 hemispheres. And this was highly significant. The next stage was, well, we have this pattern, we have the network expression, but does this biologically make sense? And does the topography fit with what we know about STN DBS? It's been roughly about 35 years since the first DBS procedure for PD was done. The initial target was VIM, followed by the STN very shortly. And so we have a fairly good idea of what somewhat gets modulated. And the network did fit very well with the human motor system. We had significant contributions regionally from the motor cortices, the sensory motor cortices, the supplementary motor area. There was some modulation of the prefrontal cortices and the cerebellum, which plays a very big role in tremor modulation. And then you also had significant contributions from the STN, and there was some seeping through into the GPI, which is in close proximity, which also showed significant modulation. So biologically, this made sense to us. And the reason why we chose a hemispheric pattern is you may have an electrode appropriately placed in one hemisphere, but may not be well placed in the other hemisphere. So you may not see the same biological effects in both, and the human motor system is sufficiently lateralized to be able to separate the two out. So what does it mean to have a biological network that is associated with the treatment response? Is it just simply an anti-PD network? The answer is yes and no. In the blue and the red, you can see the standard Parkinson's disease-related pattern, and in the yellow, you can see the significant regions that contributed to the STN StimNet network. And you can see there's some amount of overlap. There's a lot of regions that may be more associated with the cognitive effects, like the prefrontal cortices, that don't really show up on STN StimNet, but you do see these regions show up, the motor regions show up between the STN and PDRP, between STN StimNet and PDRP. What's interesting, a lot of these regions also show the opposite effects compared to what you would see in PDRP. So when you have a higher expression of PDRP, the sensory motor cortex typically is red, versus in STN StimNet, the sensory motor cortex goes blue. The cerebellar vermis is red in PDRP, it's blue in STN StimNet. So in a way, you can see that there's at least some degree of modulation that's going in the opposite direction of PDRP. So far, everything biologically checks out. It kind of makes sense that patients, when they're on, they don't tend to have the symptoms of PD as much, and that kind of fits well with the PDRP notion. But what's the use of a network like this? Is the network specific to the treatment responses with STN stimulation, or is it kind of every treatment in PD gives you the same response? So for that, we did a couple of things. We wanted to first look at the stability of STN StimNet measurements. If I were to scan the same patient days apart or weeks apart, would I get a stable STN StimNet score? And that is true. You can see the column on the left-hand side. It has a pretty good repeatability. We then looked at the modulation of STN StimNet when you only gave levodopa. These were 10 separate patients not involved in the STN StimNet derivation. And you can see that with levodopa, STN StimNet is not really significantly modulated. The expression pattern looks a little bit messy. When you take DBS interventions such as the GPI and STN, you now start to see modulation of the STN StimNet network. It was more so and a little bit stronger for the STN cohort, but I don't think with the small group, we could conceivably say that the GPI was significantly low in terms of its modulation. And finally, what was really interesting is prior to the advent of DBS, we were routinely doing lesioning procedures, and the subthalamotomy had become particularly popular. The problem with the subthalamotomy is it was usually restricted to just one side because the side effects were pretty high. This becomes particularly interesting in the modern era of HIFU, which is a non-invasive lesioning, and people have spoken about potentially performing the subthalamotomy again for these patients. And you can see that when you perform subthalamotomy, which may be mechanistically different, even though patients have the same kind of benefit, network expression actually went in the opposite direction. StimNet expression actually drastically decreased. And this kind of makes sense, what DBS does to the STN versus what lesioning does, but this was interesting. We compared the subthalamotomy group to a sham surgery group, which was patients that were part of a clinical trial but had no intervention and were scanned across a year just to compare the subthalamotomy group to the sham surgery group, especially given the interval in scanning was 1 year. So it biologically makes sense, but does it correlate clinically with the improvements patients experience? Now remember, STN StimNet was not derived with the motor improvements fed into the algorithm. It was purely derived on the separation of the off and on condition. But when we correlated the changes in the StimNet scores, which you can see on the x-axis, it very strongly on a hemisphere basis as well as on a whole brain basis correlated with the improvements on the UPDRS path 3. This also held true on the whole brain portion of the comparison when you start to factor in the axial symptoms that patients experience that get taken out when you look at only limb scores. Obviously, these were all patients scanned from the same scanner that StimNet was derived in, so we wanted to validate our findings, and we had our collaborators from Italy that were kind enough to provide a data set that had previously been published after STN stimulation on and off. And you can see that when you compare the changes in StimNet expression, this correlation also holds true on a completely independent data set. The next thing that I think was very interesting was, is there a dose-response relationship? We know that as you increase stimulation, symptoms tend to get progressively better. There is a ceiling effect that we'll talk about in a second, but they seem to get better. So to analyze this and to probably study this a little bit better, we looked at O15 studies, specifically because they have a short half-life and we can sequentially scan them at different stimulation parameters in the same day. And the first thing we want to do is StimNet measurements on FDG and O15 similar and stable? And we can see it has a very strong correlation when scanned on the same group of patients. But what really got us interested is that when you take the voltage, that is the amount of stimulation being delivered to the STN, you see that there is a positive correlation with the amount StimNet changes. So that makes sense. The more you stimulate the STN, the more you expect STN StimNet to change. But what if you did that on a patient-by-patient basis? What if we just didn't average the group? So this is just a figure from a software called DBS that allows you to model the electrodes and its relationship on a patient to the STN on a patient-by-patient basis. When you turn the stimulation on a subtherapeutic basis, subtherapeutic is defined as the threshold at which tremor reemerges. The reason we use tremor because it tends to come on and off pretty quickly with stimulation versus some of the other symptoms that take a little bit longer to respond. And you can see that the volume of tissue activated, which is in this red blob here, is within the motor part of the STN that we want to stimulate, but it's not too big. When you reach the therapeutic amplitude, this is the amplitude that the neurologist had determined to be most optimal for patient control. You see that this VTA is a little bit larger and it tends to modulate the fibers that connect the STN to the rest of the brain a little bit better. What happens if you run O15 scans on these three conditions? And we actually found a positive dose-response relationship where we see a separation between the STN stimulation on and the therapeutic amplitude, but we see a progressive increase in the subtherapeutic amplitude as well. But we know that it's not something that you can progressively increase. If you increase the voltage too much, patients start having side effects. They can have dyskinesias, they can have capsular side effects, they can have sensory disturbances. So does StimNet just keep increasing as you increase the stimulation? And so we again scanned a small cohort of patients in three conditions: the off condition, the on condition, and then we scanned them in a super on condition or a super therapeutic condition. And the super therapeutic was defined as the lowest amplitude at which patients start experiencing dyskinesias. And these dyskinesias can be pretty troubling and painful to patients. So, we had a much smaller cohort of patients that could do it and tolerate it. It was for a brief period, but it also gave us an insightful knowledge. When you go past this therapeutic amplitude, StimNet expression starts to break down, starts to go back to baseline levels, and even starts hitting levels lower than what they were before, suggesting that StimNet is capable of finding this dose-response relationship. And then, one of the final parts of the biological validation that we performed was, does the StimNet expression is fundamentally based in the physiology of the STN? So for that, we took a cohort of patients that were prospectively enrolled for the purpose of the study. They underwent resting state fMRI scans preoperatively, and they all underwent DBS implantation with an MRI compatible stimulator, but one that also was able to record local field potentials in real time outside the OR. So it could be somewhat compared to the conditions in which the resting state fMRI was performed. And for the purpose of the study, the first thing that we did is we measured the low beta power. Now, low beta is very well known as a marker of symptomology in PD, and it correlates very well with the motor disability that patients have. And first, we wanted to validate this approach. And you can see that beta correlated very well with the motor symptoms that patients were experiencing. What was interesting is that beta did not correlate with StimNet expressions. So, it got us thinking, does that mean that beta, which is a pathological marker of PD, may not correlate with StimNet? Because StimNet is not inherently a pathological marker of PD. It's inherently a marker of an STN-related network. We then looked at the theta activity in STN StimNet, and theta is defined as 5 to 8 hertz. And theta has been described by groups as being very relevant in interregional communication and one that drives synchrony. And so when we correlated the STN theta power, we found a positive correlation with the STN StimNet expression, suggesting that the STN was really driving the activity of this network in the off condition. And this has been shown before. There's a very nice paper in I think Nature Communications that was published a few years ago that showed that the way the brain chooses to communicate across regions is in this theta bandwidth, and gamma represents more local spike in a local activity of neurons. We took it one step further. We looked at the STN theta activity and looked at the localized FALF values for different regions that we know are important in StimNet. And you can see that regions that typically are functionally connected to the STN that are part of StimNet had very strong connections with STN theta activity, especially for the GPI, suggesting that the STN theta activity was driving the activity within these regions as well. And then finally, we wanted to topographically verify, is this network and the nodes that contribute to it anatomically and physiologically truly related to the STN? So for the purpose of this, we took two patients and we placed, during surgery, we placed subdural electrodes along the cortex relevant to motor regions, and simultaneously recorded LFPs from the STN as well as LFPs, as well as ECOG from the cortical regions. The cortical regions are depicted in this figure here. You can see that they are color-coded based on what the region weight on STN StimNet was, just to give you a sense of what that would look like. And then we calculated two things. We calculated whether when you turn stimulation on, is the neuronal activity within these regions match up to what you see on StimNet? And this was done by measuring the finely tuned gamma activity within the cortical nodes. And you can see that regions that had very high region weights on STN StimNet, they also had extremely high modulation of gamma activity. And this relationship went both in the negative and positive direction, suggesting that regions that were thought to shut down with stimulation did in fact have lower gamma activity, and the other way around as well. And then finally, to kind of hit home the hypothesis that is theta that drives this gamma activity, in these patients we measure the modulation index, which is how much of theta activity in the STN drives the gamma activity in the cortex. And again, this was only 2 patients with about 15 contacts. But you can see that there is a relationship suggesting that the amount an area responds is in fact related to the modulation index that we measured intraoperatively, suggesting that STN StimNet is very fundamentally based in the physiology of the STN. So all this is well and good. We validated the biology of STN StimNet, and I kind of want to get into the main crux of this topic is how does this help us clinically? So this really came about by observation that just something that we looked at was that the off stimulation measurements of STN StimNet correlated very well with the motor improvements that patients experienced with stimulation, and this was true both at our site as well as site 2. Again, these are off scans, they're not truly a preoperative or a baseline measurement. So, one can only make so much of an inference from this. So the next step we did is we took 12 patients who had undergone FDG PET scans roughly about 8 months prior to surgery. And they had motor assessments done about 17 months after surgery. And all these patients had undergone FDG PET. On top of that, we also had our resting state fMRI cohort that were the 15 patients prospectively enrolled, and they'd undergone the resting state fMRI as part of their pre-surgical workup 12 days prior to surgery, and they underwent motor assessments 5 months after surgery. And 5 months was chosen at the time when the neurologist felt on average they were somewhat stabilized on their stimulation parameters. And you can see that we again see this relationship. This relationship holds true where the preoperative scans were able to predict how much benefit patients were able to experience. So that's massive. This allows us to now start thinking about moving past the levodopa challenge test, which, a lot of neurologists have spoken to have also said that, it's somewhat of a guide, but it's not very objective in how much benefit a patient would experience. So first things first, the baseline StimNet expression did have some correlation with the levodopa challenge test. It was not very strong, but it was there. But does that just mean StimNet is capturing what levodopa challenge test can do? Or was that something different? To better understand this, we performed a multiple regression analysis on the resting state fMRI cohort. And the purpose of this was not to create a model that is predictive, but to look at what the contribution of each of these factors were. We also put in the disease duration, which is known to influence the levodopa challenge test, as one of the variables. But you can see that the beta coefficients for STN StimNet were far stronger than LCT or duration. And LCT almost becomes non-significant when you enter StimNet into the model, suggesting that StimNet is absorbing all the variance from LCT and more. And you can see that with some of the partial correlation plots on the right-hand side. This was also seen by the Shapley scores that you can see in red next to it, suggesting in the different permutations, StimNet does hold up as potentially being superior in some ways to the LCT. The last thing, we know that electrode implantation matters. So what if all these changes that we're seeing in the resting state fMRI was simply because they had good electrode placements? And these questions did come up when we were discussing this. So we modeled the electrode location of each patient bilaterally. And we know that the overlap within this area that is highlighted in green, the green blob, which is considered the sweet spot of the STN for stimulation based on work that was done by Dembeck. I think it was published in 2019. But you can see that the amount of the overlap, which was measured by the Dice coefficient with this sweet spot, did correlate with the amount of improvement patients had. Okay, so electrode location matters. We validated this. That makes sense. But when you combine that with preoperative StimNet scores, just to highlight the potential of how we could use this, you can see that the prediction becomes far superior. We have an R-square of 0.53, and StimNet continues to remain a significant contributor even after controlling for electrode placement. And so If we've sorted out surgical accuracy, identifying the correct patients will potentially help us even better understand who is a good candidate. And then finally, one of my favorite slides of the entire project in the paper was, how do we use this in the real world? So to do that, we wanted to see how would StimNet function and move from just a biomarker to helping candidate selection. And we looked at a large cohort of PD patients across the entire lifespan of Parkinson's disease. And the first thing that I just want to highlight is that when DBS first came out, it was these patients that had been diagnosed for at least about 8 years in the advanced PD group that were typically candidates. As we've gotten better at it and experience has gotten better, once patients start hitting this 4 to 7 mark, we start to think of them as potential DBS candidates, especially if they're not able to tolerate the medication or not getting adequate relief. So we calculated StimNet scores in all these patients. We also took a cohort of patients that were drug naive, that never been exposed to dopaminergic medication. So by definition, would probably not be candidates for DBS. We also took some RBD patients that may have phenoconverted in the future to PD, atypical Parkinsonian syndrome patients, and there's healthy controls. And you can see that there is a stepwise change in STN StimNet values across the lifespan. But you see that this starts to plateau out at around the 8-year mark, which suggests that maybe if we were to validate this in a larger cohort, there's no real benefit in waiting to get to this advanced stage. The benefits that you get with STN DBS may be sufficiently, plateaued by the 4-year mark. So maybe patients can be enrolled earlier for surgery and maximize the years of benefit that they get with DBS. But then how do you determine who is a candidate, who is not a candidate? So just for the sake of a thought experiment, we wanted to see that if we take something called the clinically important difference, and this threshold in large studies has been found to be about 10.8 points on the UPDRS Part 3. This is called the large CID. And when we're talking about a surgical intervention, you typically expect to want to have a large CID. And patients would typically not be keen if they were more in the small to moderate groups that may be sufficient for medication trials. And you can see that meaningful response was predicted once you hit that 4-year mark. Again, lines up with what we anecdotally already know, but has not been proven in the literature. And you can see that the number of patients that would qualify steadily increases and starts to plateau again around the 8-year mark. Patients that would have otherwise not been candidates for DBS, atypical Parkinsonian syndromes, never meet the threshold for a large CID. Same with REM behavior disorders and same with drug-naive Parkinsonian disorders. This kind of gives you a sense of what this may look like in the real world, but also helps you validate in the clinical intuition that we've had so far. Too early PD patients may not be optimal candidates, and patients that are too advanced have probably already plateaued their maximum benefit, and we could have probably done this a few years sooner. And last to kind of really drive on the topic is we know we can calculate PDRP expressions. We've shown that STN StimNet is more specific for STN DBS. So if we were to do the same experiment with PDRP expression scores, you do see a difference, suggesting that StimNet has a biology and behavior along the lifespan of PD that is different from PDRP. In PDRP, you see a linear, gradual change in PDRP scores. Just off note, PDRP scores had been inversed for the purposes of display because I just wanted to have a fair comparison with PDRP and not to confuse. PDRP typically rises with disease duration. So we just transform the data. But you can see that while PDRP has this linear trend with disease progression, you don't really see that with StimNet. And you can see the trajectories are very different in the small inset that you see at the bottom. So how would this work in a clinical workflow? This is just a schematic diagram to kind of highlight and drive home the point. Let's say you have a patient that's coming in for evaluation for STN DBS. You could either scan them preoperatively with PET imaging, or you could just scan them with MRI. All patients have to get MRIs anyway for preoperative evaluation, and you could easily do this in resting state fMRI. And you measure the StimNet expression on these patients, and you see whether they fall within the threshold of CID that you and the patient have determined to be what will be within their goals and their expectations. If they were to fit this criteria, that's one piece of information that goes into the multidisciplinary meetings that most major centers have where neurologists and neurosurgeons sit together to discuss who's a good candidate. That's not to say that StimNet would be a sole determinant of whether someone should get DBS. There are factors like dyskinesias, motor fluctuations, cognitive status, and their levodopa requirements that all factor in. But StimNet would provide an objective metric as well as give patients the data and what the expectation of surgery would be, whether they would be getting a substantial benefit that meets the goals that they want out of surgery. Because again, I like to always talk to patients and highlight to them that DBS surgery is really a symptomatic treatment. We don't know if it changes the trajectory of PD, and most evidence right now suggests it doesn't. And so it's a very important piece of information that I think we can provide patients who otherwise are very fearful of an invasive brain surgery. So, just to drive home the point, and I think we can then open it up to questions, is that, STN DBS exerts its effect via this distinct brain network. I think this network is a biological network of the STN, not a pathological network of PD. And, we've shown some ways to kind of validate that. Stimulation increases do correlate with the amount of stimulation delivered to the STN, suggesting you could potentially even use this as a way to titrate patient stimulation parameters. There's an excellent paper from the Toronto group that was published a few years ago that showed that using resting state fMRI postoperatively in one session lasting about an hour or two, they could predict what stimulation parameters patients would have at 1 year after multiple neurology office visits. The baseline activity was able to identify the patients that are likely to experience the greatest benefit. It is suggestive of being a metabolic signature of treatment response. And then I think what is most important that I think at least I like to think about, and I'm optimistic about its utility of such an approach, is in other disease processes where we know that pathologies respond much more slowly. With DBS, you tend to see a more immediate response to stimulation, but in conditions like dystonia or psychiatric conditions like depression or OCD, that response is slower. You don't see that response in the office, so you don't really have an objective marker on whether the parameters you've set are sufficient. And so it's very important that you can maybe use a network-based approach that measures the stimulation intensity, and what that may mean in terms of the therapeutic benefit. And I think also finally for a lot of interventions that are not standard of care, such as for depression, OCD, they're still off-label uses for DBS. And when we do decide to do surgery, it's very important to identify patients that are likely going to do well for us to be able to move the needle and maybe make these treatments a standard of care. Because most large-scale studies have failed when you've gone into multi-institutional trials. There have been a couple of depression trials that failed because while small cohorts showed good response when they did that across institutions, a futility analysis showed that it was not better. But when you identify a cohort of patients that are good responders, they have substantial benefit from DBS. And so I think an approach like this could be, especially beneficial. We've been doing some similar work with depression, something that we're working on and should be having out shortly. But that's why I think this field is very, very exciting. So just to give enough time for people to ask questions, I'll open the floor up.
Thank you, Prashin, for the wonderful talk. Very thorough and exhaustive as always. Let me open the floor for the discussion, and I suggest that people just ask questions straight ahead. Or in case you can't, you can just write them in the chat and I'll read them out.
Sorry, me? Sorry, there's a very soft. So, anyways, actually, I really enjoyed that. This was great. In particular, I really like the also biological validation where you relate the loadings of the StimNet to some of stimulation. I think this seems to me it's the first time that this has been done. So it's great. It sort of exceeds the usual Heidelbergian kind of validation. So my question is, first of all, maybe you did this. If you just took, and maybe you have enough people already, if you look at the success of deep brain stimulation and just related it to the baseline or when they haven't been operated on, if you just related that to the PDRP expression, StimNet expression, as well as UPDRS, you find a good model and which one is more significant in the prediction? Maybe you haven't done this yet, or it seems to me kind of an obvious thing to try. Another question I had, I really like this also when you increased the stimulation and then saw that the StimNet pattern scores really fall down. Do you think then there's a total reorganization where the covariant relationships between the regions that you found in StimNet is no longer there and kind of the network has kind of disintegrated? What is your thought on this? Anyway, great.
Thank you. Thank you. Those are actually excellent questions. So just to start with the PDRP, I didn't include those slides and it is included to some extent in the paper. It's I think in the supplementary section, if I remember correctly, just because it was getting too much to put in the main figures. But we actually did look at PDRP. So PDRP expression changes do somewhat correlate with the motor benefit that we see. So the deltas of both UPDRS and PDRP do have some relationship. It's weaker than what we see with StimNet, but it does exist. But we did not find any predictive capacity of PDRP in terms of its ability to predict who's going to have how much of therapeutic benefit. And that kind of makes sense because if you think about it, StimNet tells us that there's a plateauing while PDRP expression continues to increase as the disease progresses. So I'm assuming that those moderate to advanced disease patients, PDRP scores may be disproportionately high, but the amount of benefit they are experiencing has kind of already plateaued out. And so we didn't really find any relationship with the baseline scores of PDRP and the delta UPDRS. And with the next question that you asked about the stimulation, and I can maybe just pull up that slide quickly. So I have a couple of thoughts of why that may have happened. When you do increase the stimulation beyond what is therapeutic, I do think there is probably some component of the network disintegrating. I don't know whether that just completely shuts off the STN beyond what we wanted to. But I do think the more likely scenario is that you probably have spread of current into the associative and limbic parts of the STN. So you start having distant cortical areas that may not be related to StimNet now starting to change their biology or their firing. And that probably throws off the pattern. Additionally, I think more likely also is going to be the spread of stimulation to the internal capsule. So while you may start seeing decreases in the sensory motor cortex with StimNet when you're in the STN, but when you directly stimulate the internal capsule, maybe you see a reversal and the sensory motor cortex actually starts to become more red or more active because you have antidromic stimulation going back to the sensory motor cortex. So I think it's probably a little bit of more of stimulation spread that's not specific to this signature pattern that does correspond with the therapeutic benefit.
I could ask. I have even more questions. I don't want to hog the floor and crowd people out, but maybe give some other people the chance to speak. But
Hi. Yeah, I have a question. I don't know this too much, but I'm thinking about, so this is a Parkinson disease and usually it is related to dopamine, right? So you are using FDG and water to measure blood flow and metabolism. Have you tried, or do you think that using PET to study dopamine would be a better way or a different way?
That's a topic that I've been interested in. I think the utility is low because the reason being is we don't really stimulate the substantia nigra, which is what's degenerating in Parkinson's disease, and that's where the dopaminergic deficit comes in. We're stimulating the STN, and I had a slide that maybe I can just bring up that may so this figure, which is the indirect pathway that kind of we all know is that the STN tends to become hyperactive in Parkinson's disease patients. So when we put the DBS electrode, we're not really combating the dopaminergic deficit. What we're really combating is somewhat shutting down the hyperactive STN. And what that downstream does is it reduces the excitation to the GPI, which then releases the brakes on the thalamus to then go ahead and stimulate the cortex. And maybe that's why they become less bradykinetic. That's why maybe the rigidity goes away. So I don't know if we'll actually see enough signal with dopaminergic related markers with PET. But it would certainly be interesting. I think in depression, I've actually been very interested in that because one of the targets that we use at our institution, which is one of the only two places in the world that uses the target, is this area called the medial forebrain bundle. And that tends to connect to the ventral tegmental area, which supplies dopamine to the prefrontal cortices. And I suspect that in that disease process, stimulation may potentially see some changes in dopaminergic activity, especially if you measure them across, a time span. So if you measure them at baseline and then after 6 months or 1 year, I suspect that you'll see dopamine binding maybe changing in the nucleus accumbens or the frontal cortices. But that's definitely something that I have thought about conceiving. But I don't know about PD, just because I don't think we're in any way modulating the dopaminergic pathway with our stimulation.
Let me ask you a question, Prashin.
Yeah.
I know David has been interested in this a lot, and in the slide that you have shown before, you have the sham surgery patients. And we see this placebo response when in some patients. Have you tried looking into if this placebo-related pattern can give you additional information on the top of the StimNet to maybe even better find who is the most appropriate candidate for the DBS?
No, that's actually a really, really good point. We kind of spoke about it a little bit, and then I think it just, in the series of experiments that we ended up doing in the analysis we ended up doing, it kind of fell off sideways. But I do think that's very, very important. And I think it's particularly important to add that in because I do think there is some amount of placebo response, especially because these patients are not just getting surgery and then they're kind of done with their treatment. They constantly are seeing the neurologist, constantly getting treatment. And I think that potentially reinforces some degree of placebo response because they have constant medical attention. So, I do think that may help a little bit. I think we were kind of limited in the number of patients we had. So any sort of, regression model that we keep adding variables to, we were kind of limited in what we can do. But yeah, I think for sure, I think there would be something to think about the placebo response, definitely.
Hi, can I ask you a question? Hi, Prashin.
Hi.
Congratulations for a really interesting talk. I have a few methodological questions, actually. You said in the beginning that this network was identified on one hemisphere and the one that was more affected, that was contralateral to the more affected side, or that had just the best effect on DBS. Can you say it again, please?
Yeah, sorry.
How
So
did
yeah,
you
maybe
how did you choose the hemisphere?
Yeah, absolutely. So yeah, maybe I didn't clarify that well enough. So what we basically did is, initially we thought about potentially just doing a whole brain network, but we found that the effects of stimulation are sufficiently lateralized. Their stimulation parameters are different on each side. You turn one stimulator on, you only see effects on the contralateral side. You don't really see effects on the ipsilateral side. So we know that they somewhat function distinctly, and I think it would be more helpful to have a hemispheric pattern. So for the purpose of this, what we did is we took our entire cohort of patients from North Shore and we divided them based on the hemisphere and we took the best performing hemisphere. So ultimately, I think all the patients that were included in this derivation cohort were from the same 5 patients, but we identified the network on a hemisphere basis. And we did that. We selected the ones that had the most improvement on the UPDRS. So while we didn't feed the actual UPDRS scores or the magnitude of change, we did select the best performing hemispheres to identify the network because we just felt that meant that we're probably modulating a network that would be of interest to us. And that's how we kind of narrowed in on the cohort for derivation.
And that was not necessarily the more affected hemisphere, right?
Correct. Not necessarily the most affected hemisphere. It was purely based on the delta.
And I have another question. Do you do any follow-up about on these patients, what's happening with the StimNet on a long run on these stimulated patients? Does it change at all with disease progression?
We somewhat tried to answer that with that final slide that I showed with terms of, the lifespan. Obviously, it was not within subject. It was kind of, a large cohort of patients. But we haven't done that on patients because it's a little bit challenging to get them to come for follow-up scans. What we are working on right now is, I think for clinical applicability, it's a little bit easier to get patients to do a resting state fMRI sequence just because the MRIs are kind of part of their care. We sometimes will get MRIs postoperatively just to look at the electrode location so that we can identify what contacts to use for stimulation. So some of these patients are getting scans at like 6 months and then 1 year. And that's something that we'll probably look at, but I don't know if we'll end up having enough power, at least in the near future, to make any sort of determination. But I think that's a valid point, does the StimNet kind of plateau out? But one of the confounders that, as I'm talking, that kind of hits me is that the stimulation parameters don't stay static. So whether those changes in StimNet, if we do see any, at 6 months, 1 year, or 2 years, are they because of disease progression or are they because of, the stimulation parameters changing? Now, you could scan them in the off condition, but we've already seen that at 1 year, StimNet scores remain, relatively stable with the sham surgery group. You see that there's not a lot of change when I showed that slide, that there's not a lot of change at 1 year in StimNet expression.
Thank you.
We have a next question from Christina.
Yeah, so thank you for the very interesting talk. Maybe I missed this in the beginning. So when you do the stimulations with the FDG, is this an on-off design, like a block design? Okay, and you did the same for O15 water and FDG because you showed this very high correlation between the two measures. How would you interpret this?
So yeah, it was a block design. It was patients were scanned on 2 consecutive days, and I think this is the slide. So they were scanned on 2 consecutive days. On day one, they came in the off medication, off stimulation phase, and they got an FDG scan. The next day, they came in the off condition scan, and they had an O15 study done in the off condition. We turned the stimulator on, waited about an hour or two till their symptoms kind of plateaued in terms of improvement. This was done through serial UPDR scores, and then we landed up scanning them in the on condition with FDG PET at the end of that day. The reason we did
but between on and off FDG, that was not within the same scan, was like two separate scan, one with on condition and one with off condition, right?
Correct. Because we couldn't, because of the half-life of FDG, having to wait enough time, we kind of separated the FDG scans onto 2 separate days because otherwise they'd have to wait too long, to inject them twice in the same day. So we did stagger them for the FDG scans.
Okay. And the correlation that you see between O15 and FDG, what's your interpretation about this?
So the point of doing that was one, to quote Dr. Eidelberg is, which Matej will find is what gives you the right to measure an FDG related pattern on O15, as he famously told me is, we wanted to see whether measurements on FDG and O15 are compatible. And now if intuitively they should be because O15 measures blood flow is coupled to metabolism and, you should see it. But I think the purpose of that was just to show that yes, when you measure the same candidates on FDG or O15, especially the deltas of the StimNet are highly correlated in either condition. So that was more to kind of prove that our O15 experiments were valid and, meaningful versus someone saying that, oh, but, it's a completely different marker and it's a network derived out of FDG, not O15. So that was really the purpose. And I think it kind of held up pretty well.
Okay. Thank you. And maybe one more thing would be like, deep brain stimulation in the STN has been along for a long time. And what's the current status of research? Which regions are, besides that nucleus, interesting? What do you think would be other interesting regions to explore for these kind of stimulations?
Yeah, that's a great question. For PD, I think we've kind of plateaued on trying to stimulate something else just because in general patients respond so well to stimulation in these conditions, and mechanistically it kind of intuitively makes sense. So it's really the STN and the GPI. There was some interest in stimulating the PPN, especially for patients that had freezing of gait. That doesn't get well treated with STN or GPI DBS. But these typically hold very true. We've kind of moved away from stimulating the VIM for PD patients because VIM only really helps in tremor because it modulates the DRTT pathway. So it's really reserved for patients with essential tremor. We don't really do that much for PD. But for other diseases, I think there has been a lot of interest. For example, with tremor, people have been talking about stimulating the posterior subthalamic area. It's kind of an area where the DRTT traverses after it leaves the VIM going towards the cerebellum. And that has shown like very good benefit. There's an area called the zona incerta, which seems to have extremely good benefit, and people have postulated using that for DBS as a target. But I think what's exciting is in conditions outside of PD. I think in depression, there's so many areas that are being stimulated. The subgenual cingulate is the most common. People have done the anterior limb of the internal capsule. The ventral striatum tends to be a good target for especially addiction. I think there was a very nice study that came out in Nature Medicine this year by Casey Halpern's group where with GLP-1s being in vogue, they had patients who were being treated with nucleus accumbens DBS for binge eating disorder, and they actually found GLP-1s blunted the pathological response that is associated with binge eating, kind of giving mechanistic validation to why that may be a good target for binge eating disorder. So I think that's really where the exciting stuff is. For depression, I think what I'm most excited about is an area called the medial forebrain bundle, and that's an area that's a little anterior to the STN, and it was actually found in PD patients because PD patients that had DBSs that were misplaced too anterior into the STN became hypomanic when they were stimulated, and people were like, "Oh, why does that happen?" And I think one of the notions was that, "Oh, you're stimulating the medial forebrain bundle, and maybe hypomania in the right patient could potentially lead to some degree of antidepressant effects and reward seeking." So, that's kind of where it came about. So I think that's probably where the exciting stuff lands. I think with PD, it's more about optimizing the outcomes, making things more reliable, and also just how can you quickly optimize these patients. I think a lot of the research is in how do you optimize stimulation parameters. So I think that's where most of the focus right now is.
Yeah, thank you very much.
Thanks, Prashin. Maybe we have a time for a very quick last question and a quick answer. If there is any burning questions from the audience, Chris.
Yeah, I could. I'm gonna ask a bigger question over email, but just regarding the StimNet obtained from the 015 and the FDG, you showed that the stimulation-related pattern score increases were highly correlated. The pattern itself, does that look topographically similar also, or is it quite different? Maybe you showed this, but I might have missed it.
So we didn't derive a pattern from the 015 studies. We tried to do it, but I think 015 is a little bit noisier to actually be able to reliably derive a network. What we did is we used the FDG pattern and then calculated the expression scores on the 015.
Prospectively applied.
Yeah.
You
Yeah.
just prospectively applied.
And really the goal of the 015 was really just to validate this stimulation dose effect, and that's really the purpose of it. But yeah, I think maybe it'll look similar, maybe it'll look different. I'm not sure, but I would suspect because they're correlated, they would look very similar.
Alrighty.
Thanks.
I had a question about ergodicity because that's our obsession in this group, but I asked it over email. It's a long and rambly question. Anyways, great talk.
Absolutely. Thank you so much.
Thanks again, Prashin, for the wonderful talk, for the wonderful discussion. And I think, for the sake of timekeeping, I think we can close the session for today. Sheen posted 2 links in the chat for joining our next online series, and this seminar will be available on YouTube shortly. Thanks, everybody, for joining in today.
Thank you, everyone.
Bye.